Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type your questions in for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Kajal Brumbat, Director of Quality Control Analytical Testing for WuXi Advanced Therapies.
Hello, everyone. My name is Kajal Brumbat. I'm the Director for QC Analytical Testing at WuXi Advanced Therapy. Today, we'll be discussing about best practice in mycoplasma testing for cell and gene therapies. Today's webinar, we will be covering three topics. The first one is mycoplasma detection, the importance of robust mycoplasma testing for cell and gene therapies. With that, the next topic will be mycoplasma detection methods. What are the methods that are used to detect mycoplasma with their advantages and disadvantages? With that saying, we'll also go a little bit more into what services are offered by WuXi Advanced Therapies to support the mycoplasma testing. So mycoplasma detection. Before going into that, let's first discuss about what is mycoplasma. Mycoplasma is smallest bacteria, variable in shape, and unlike other bacteria, they do not have a cell wall. These are common contaminants in biopharmaceuticals and therapeutic industries. The source of mycoplasma are basically plant and animal tissues, and hence the processes that uses or involves these sources are prone to contamination by mycoplasma. Let's discuss about mycoplasma contamination and its impact. As mentioned, mycoplasma is a common contamination seen in cell cultures. About 5 to 35% of cell cultures worldwide are contaminated with at least one species of mycoplasma. Again, mycoplasma contaminations are not easily detected and does not have any visual signs of contaminations, which makes it more harder. These contaminations are hard to detect, unlike other bacterial contamination, which are visible through turbidity or change in their pH. With the fact that mycoplasma contamination are harder to detect, if they are not detected early in the process, it can pose significant economical and safety risk, which may result in compromised product quality, loss in production time, patient safety concerns, also loss of product, which eventually will delay in providing therapy to patients in need. Mycoplasma contaminations are also direct and indirect causes of human disease. Due to the lack of cell wall, they are resistant to beta-lactam antibiotics such as penicillin. As discussed above, how mycoplasma contamination is very impactful and can have a significant impact, it becomes more important to test the mycoplasma earlier in the process. Let's discuss about the importance of mycoplasma testing. Performing mycoplasma testing at the right time, it ensures the product quality and patient safety. It also ensures smooth productions. Again, as mentioned, right, in appropriate quality control testing for mycoplasma throughout the manufacturing process is critical to avoid any significant impact on the production and the product purity, safety, and potency. Based on the criticality and importance of this mycoplasma testing, it is very important that this testing is performed at the various phases of product development. Ideally, and also recommended by regulatory agencies, this testing should occur at a raw material level, where it is important to ensure the raw material used for the processes are mycoplasma contamination free. It also needs to be tested for cell banks, all unprocessed, processed, material used for the drug substances or drug products. And of course, it's important to test the mycoplasma at the final product. As discussed above, right, how mycoplasma contamination can impact the product and also the patient safety. It is now important. It is also important that we have a strong, robust method to detect the mycoplasma. In this section, we'll be discussing about the mycoplasma detection methods that are offered by WuXi Advanced Therapies. WuXi Advanced Therapies offer following three methods to test for mycoplasma. Number one is gold standard method. This is a traditional method that uses a microbiology culture method. The second one is a touchdown PCR method, and the third one is a MycoSeq rapid PCR method. As the name suggests, the last two methods are PCR-based method. We at WuXi Advanced Therapy offers a gold standard method, which consists of two parts, direct and indirect assay. This method is a regulatory compliant method and has been approved, universally approved and accepted by following agencies. The method aligns with USP, EP, and we also offer another method which is aligned and aligned with all three pharmacopoeias. This method can test for cultivable mycoplasma strain, which uses a direct assay part of the method. It tests for a non-cultivable mycoplasma strains, which is detected using indirect assay. It also tests for inhibitory substances in the samples, which is also called as mycoplasma stasis. And lastly, the method can also test for spiroplasma, which is also suggested by agencies to be tested for biopharmaceutical products that have been in contact with plant and insect material. With that, let's go into a little bit more detail on how this gold standard method has been executed over here at WuXi Advanced Therapies. As mentioned above, gold standard method consists of two parts, the direct assay and the indirect assay. Let's talk about the direct assay. The direct assay is a traditional culture method that takes about 28 days for its completion. Um, the method is basically divided into two portions where the sample, and if you're testing for mycoplasma stasis, the sample is spiked with the controls and along with control are tested and inoculated onto a broth, which is a liquid media, and on the agar plates, a solid media. Following the flowchart as seen on the screen, the broth is incubated for about 28 days throughout the assay time. And during the incubation period, the broth is observed every 2 to 3 days for any changes for potential mycoplasma detection. Um, the broth is also subcultured at certain time point on the agar plate to identify any potential mycoplasma growth seen during the process. The agar portion of the assay, the plates are incubated for 14 days and then read for any potential growth for mycoplasma colonies. At the end of the assay, on day 28, a final read is performed um, where the results of the sample are compared with the control to identify any potential growth of mycoplasma. Now, as seen on the screen in the picture right side, the first picture shows the broth observation for mycoplasma positive controls, where the color differences will indicate if there is any growth for mycoplasma. The pink one shows the sample or a negative control with no growth versus if there is any potential mycoplasma, you will be seeing a change in a color, which may be orange, or if it is extensive growth, it will be yellow. The bottom picture also shows the microscopic view of mycoplasma colonies, which can be seen if there is a presence of mycoplasma on the agar plate. Again, as mentioned, this assay takes about 28 days for completion. Let's get into the second part of the gold standard method, indirect assay. This assay takes approximately 7 to 8 days for its completion and is used for detecting non-cultivable Mycoplasma species. The sample for this method, the sample and control, and if testing for Mycoplasma stasis, the samples are spiked with controls. All of them are inoculated on indicator cells. Vero cells are usually used as an indicator cell for this assay. Following the inoculation, samples and controls are incubated and subpassaged on day 3, followed by a fix and staining process where DNA are stained and slides are read for detecting the presence of Mycoplasma. The pictures on the right indicate the Vero cell, which is seen in a fluorescent color, um, and the first picture shows the Mycoplasma species presence. With that, this slide shows an overview of the gold standard method, which covers both indirect and direct assay. As seen on the screen, gold standard method takes about 28 days, which covers indirect and direct assay. Following the assay completion, let's talk about its data analysis and interpretation. As mentioned, the test method can detect the presence of Mycoplasma. This is determined by comparing the results obtained in the broth as well as on the agar plate with controls and if there is any potential growth seen. Same is followed for Mycoplasma stasis. This is determined by appropriate recovery of spike control organism in presence of test product. Data interpretation for this assay is very straightforward, where direct and indirect assay readout is presence or absence of Mycoplasma as well as inhibitory substances. Now that we have seen how the gold standard method is executed, let's talk about its advantages and disadvantages. Advantages. For the gold standard method, this method is considered as a robust method to test the Mycoplasma. It detects the presence of inhibitory substances. It works for both rapid and slow growing organism in broth. There is no false positive. It can also be combined with spiroplasma testing. This method can test both cultivable and non-cultivable Mycoplasma. The method is universally accepted and approved by regulatory agencies and is ideal for lot release testing. Now, along with advantages, this method also has some disadvantages, which number one, of course, it takes about 28 days to complete the assay, which is a good long time. Along with that, because it takes this long time, it's not suitable for in-process testing. That is also one of the critical part of the Mycoplasma testing process. With that saying, let's move on to the second method that WuXi offers. This is a touchdown PCR method. This testing is offered by QC Molecular Biology Testing Service here at WuXi Advanced Therapy. Mycoplasma touchdown PCR method workflow is seen in the slide here. It's an easy 4-step process where initially DNA isolation occurs through a manual extraction process, which is followed by DNA quantification. Once the DNA quantification is completed, it goes through a PCR process, and endpoint PCR is then tested using a gel electrophoresis method. This method is traditionally a PCR-based method, as the name suggests, and has its own advantages and disadvantages. Let's talk about the advantages. This method takes less than 7 days for its completion, and since it is less than 7 days, it can be used as an in-process control. The method is approved by European Pharmacopoeia. Let's talk about the disadvantages of this method. It can be very subjective, and it's very open to interpretation at the limit of detection. It may read false positive, and it cannot detect the presence of inhibitory substances like the gold standard method, as we discussed above. Let's talk about the third method that is being offered by QC Molecular Biology Testing Services at WuXi Advanced Therapy here. That is MycoSeq Rapid PCR. This method is a PCR-based method, and it's very similar to what touchdown PCR method workflow as discussed above. The method has few differences that I will highlight here. Number one, in this method, the DNA extraction, which is nucleic extraction, is automated, unlike the touchdown PCR, which is a manual extraction process. Following the extraction, DNA quantification takes place, which is very similar to the touchdown PCR method we discussed. Once the quantification is completed, it goes through a PCR. Now, this is a real time PCR where data is collected real time and analyzed through a software, and hence the readout is through a system. This is unique and different than the touchdown PCR method, which uses electrophoresis method to read out the detection of mycoplasma. With that, let's discuss the advantage and disadvantage of this method. The advantages, of course, it can be used in process control since the method takes less than 1 day to complete. This method has been approved by European Pharmacopoeia and can be completed. The assay can be completed within 1 day, as mentioned. Along with advantages, this method also has some disadvantages. It has a limit of detection. It has very restricted regulatory need. This method needs to be validated prior to its use, and it cannot detect again presence of inhibitory substances like gold standard method. As mentioned, WuXi Advanced Therapy offers three mycoplasma testing methods discussed above. Number one, gold standard method. This is a culture-based method. It's also universally accepted and approved by all agencies. It's a traditional method that is used to detect mycoplasma. The second one that we discussed was the touchdown PCR method using gel electrophoresis. And the third one is a MycoSeq mycoplasma detection system, which is again a PCR-based method. We also offer validation and verification services based on client-specific needs. Let's quickly discuss about the mycoplasma testing that is offered by QC Analytical Services, which is a gold standard test method. The table as presented on the screen shows few test methods that are offered by WuXi Advanced Therapy. The last column describes the organisms that are used as a control for those specific test method. For any special request or additional mycoplasma testing need, please contact our BD or CD teams at advancedtherapies.com/sales. These are the few of the methods that are offered currently at QC Analytical Services. As mentioned, we also offer validation and verification services. The validation and verification of client test article using gold standard method. In this verification, client may provide us their test articles, and we use our gold standard method to perform a verification or validation of that particular test articles. We also offer client-specific test method validation, which includes any special or different mycoplasma strain that require to be validated using the gold standard method. With that, I would like to end the presentation. Thank you very much for your time.
Okay, great. As a reminder to our audience, you can go ahead and type in your questions for our speaker now. So our first question is, how much sample is needed for the gold standard method?
That's a good question. We get this question all the time. We basically require 20 ml for mycoplasma detection. If client is only interested to detect the mycoplasma presence, it's 20 ml. And if and we need 45 to 60 ml of samples if we are detecting mycoplasma along with mycoplasma stasis. It depends also on a number of organisms that needed to be tested.
And if a validation is requested for gold standard method, how many runs are needed?
Minimum one run is required since it's a compendial method. However, we can perform more based on client's request. Some clients we have seen requesting 3 runs for validation.
Okay, so it looks like we're running short on time. If we don't get to your question, we'll be passing them along to Kajal, who can follow up with you directly. Please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, can gold standard method be used for lot release?
Yes, of course. This is the method that is used for lot release. It's a compendial method and is being accepted universally and approved by all regulatory agencies.
Okay, well, thank you, Kajal. And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.